Okay, so in this last lecture, we're going to go over the second type of cell division, which is called meiosis. Now, meiosis is the cell division that we use to produce sex cells, or gametes. And of course, these gametes are going to have a haploid number of chromosomes. So we're going to reduce the number of chromosomes in the cell by half. And this is, like we've said before, this provides the basis for maintaining genetic continuity, because if we didn't cut the cells in half, number-wise, chromosome number-wise, every time the sex cells merge during sexual reproduction, you'd have double the number of chromosomes that you're supposed to have. Another big part of meiosis is that it provides genetic variation. So every time you have meiosis, the way the chromosomes line up during metaphase is completely random. So you could have, you got a 50-50 shot as to whether or not it's going to be a maternal chromosome or a paternal chromosome homolog traveling to either of the poles. So you get shuffling on that level. In addition, when the two homologs line up, the two homologous chromosomes, they actually exchange genetic information in a process called crossing over, which we'll talk about in more detail as we go forward. But this creates these kind of mosaics of chromosomes that now have both maternal and paternal genes, so that it increases the genetic variation. To give you a prime example, I thought I would just show you, this is a personal picture. Two of the kids are mine, and two of the kids belong to a friend of mine, and so there's some people that we know that we see at the beach a lot in the summer. And so just looking at these four girls, can you figure out which ones are siblings and which ones are not? You can tell all four of them really have very distinct looks. None of them look identical in any way. These two here are mine. That's Mary Catherine on the bottom and Baker on the top. You know, one's got darker hair, one's got blonde hair. They look completely different, and if you knew them in person, their personalities couldn't be any more different. These two are also siblings, and you can see one is dark complected with dark hair. Ashley over here has red hair and fair skin. They don't look anything alike either. So this is just kind of an example to show you that even though both of these pairs of kids came from the same parents, it was the same process of meiosis to produce our sex cells and the same process for sexual reproduction, we got completely different children out of it. And that is due to that genetic shuffling that occurs when sex cells are produced. All right, so because we're cutting the number down by half, meiosis actually requires two divisions, because before we divide the diploid cell, it first has to go through synthesis, replication of its DNA. So you really end up with 4 times the DNA that you need before meiosis begins. So the first round of meiosis is meiosis 1, and that's the reductional division. That's when you reduce the chromosome number by half. The second one is the equational division. This is very similar to mitosis, where the sister chromatids get separated. And each division goes through those same 4 processes that we saw with mitosis, the prophase, metaphase, anaphase, and telophase. All right, so just kind of a note about chromosomes and meiosis. So like I said, you start in the very beginning, the cell, like in the G1 phase of the diploid cell that's going to undergo meiosis. You have 1 copy of each chromosome. And then that cell is going to undergo the synthesis phase during interphase, and it's going to duplicate its DNA. And so you're going to end up with these chromosomes that are duplicated and have 2 sister chromatids being held by a single centromere for each chromosome. Now these 2 chromosomes, during the process of meiosis, the very first stage of meiosis, they're going to line up very close together and actually form what are called synapses, and we're going to talk about that shortly. But when they line up, they're so close together that when you look at them in a microscope, it kind of looks like one structure, and that one structure is called a tetrad. After the first round of cell division, after meiosis 1, these two homologs get separated. So these are like in two individual cells at the end of meiosis. So the two homologs are separated. However, the two sister chromatids for each homolog are still together. Because you still have the two sister chromatids, once the two homologs are separated from one another, they're referred to as dyads. And also you can see here and here that crossing over I was talking about, that exchange of genetic information has occurred, and that occurs before the cell division. Then, after the second cell division, you end up with the sister chromatids separating, and so this would be 4 individual cells getting produced. And each individual cell has a single daughter chromosome after the sister chromatids are separated, just like you see in mitosis. And now those individual daughter chromosomes are now called monads. And so the end result, you have 4 haploid cells being produced thanks to meiosis. All right, so that's just kind of a general overview. I'm going to show you another animation that comes with the book that will give us that same process of showing you kind of an overview of what the chromosomes go through during meiosis. And then we'll start to talk in more detail about each individual phase, the prophase, metaphase, like that. All right. Okay, so this recording is an overview of meiosis, similar to what I just described with the drawings that I made on PowerPoint. Of course, their drawings are going to be a whole lot better. So we first want to talk about the fact that before meiosis ever occurs, when you have this diploid cell, you're going to have 2 copies of each chromosome. You're going to have one that came from the father, the parental chromosome, and one that came from the mother, the maternal chromosome. And these are going to be homologous chromosomes. Now, during the first part of meiosis, the very first stage in prophase 1, the 2 chromosomes are going to line up and synapse, what we call synapse with one another. They're going to actually, based on DNA sequence, they're going to line themselves up together. And at first, you can't see the sister chromatids because the condensation of the chromosomes is kind of ongoing throughout meiosis, the first phases of meiosis. And so at first, it just looks like there's only 2, and so that's why it's referred to as a bivalent. But as prophase continues on, it becomes more apparent that the sister chromatids become visible. And so here now you can see the sister chromatids. Because the chromosomes are lined up so close together, it's called a tetrad. Now you can see here, these 2 are sister chromatids, but the 2 that are side by side in the synapse are non-sister chromatids. They're the ones that are going to exchange genetic information. And so you can see here, during meiosis, or during the anaphase 1 of meiosis 1, the 2 homologs are pulled apart from one another, and that's when they become dyads. And then the second division, you end up with the 4 monads in 4 separate haploid cells. Okay, so that is just like what I showed previously, just a little bit better animation. Okay, so now we're going to look at each individual phase of meiosis. And the first phase is prophase 1, which actually is broken down to even more individual phases, because this is definitely the longest phase of meiosis. During prophase 1, the chromosomes condense and become visible, the spindle apparatus appears, all the same kinds of things that you see happening in prophase of mitosis. But something that's very different is these homologous chromosomes actually find one another and pair up side by side. This doesn't happen during mitosis. The pairing of the homologous chromosomes and the subsequent crossing over is a very complex process, and it's actually pretty mysterious. If you read in the book and it describes what's going on, it says, you know, it's believed that XYZ, or it's theorized that this happens. You know, they still don't really know 100% that they're right. This is just kind of based on what they can see and the experiments that they've done. This is what they believe that happens, but it's still a little mysterious, the whole process. And so prophase 1 is divided into 5 substages called leptonema, zygonema, pachynemia, diplonema, and diakinesis. Now, I'm not going to expect you guys to tell me exactly what happens in each individual substage, because it's all kind of a continuous process anyway, but we're going to go through all those substages so you can see what happens overall during prophase 1. So in the beginning, in leptonema, well, really before I talk about the individual stages, the condensation process of the chromosomes where they become visible is a slow and continuous process throughout all of prophase 1. During leptonema, the pairing of the homologous chromosomes begins, and the condensation occurs in regions of the chromosome, and it actually looks like beads on a string. And you can see here in the picture, it kind of looks like a baseball, like threads on a baseball, but you can see the individual little beads. And the reason why it's slower is because these homologous chromosomes are lining up based on DNA sequence. And when they're condensed, it's hard for the sequences to interact with one another. So that's why it's a little bit slower, and you have these regions of the DNA that aren't condensed and regions that are. It's to make it easier for the homologous chromosomes to pair up. Now, when they pair up in zygonema, it's called rough pairing. So they just kind of find each other and begin to pair up. And because they haven't condensed all the way, it looks like there's just 2 chromosomes present, 2 strands. And so this is called a bivalent. And something called the synaptoneal complex begins to form between the pairs, and it's going to hold the 2 pairs together. Now, like I said, we don't see the sister chromatids yet, but in pachynemia is when the chromosomes are condensed down far enough where you can actually see the sister chromatids. And so now the chromosomes are referred, the 2 homologous chromosomes lined up together, altogether is referred to as a tetrad. The synapsis is now occurring between the 2 homologs. This is a very intimate pairing. When I described what was going on before during zygonema, the bivalent, I think let's see if I can write this, the chromosomes were like 300 nanometers apart, which is fairly small. That's terrible writing. But in pachynemia, they're only 100 nanometers apart. So they've gotten much closer together. You know, a nanometer is a billionth of a meter. So we're talking about, you know, actually more than that. Yeah, it is a billionth, correct. Yeah, it's a billionth of a meter. So this is teeny, teeny, tiny spaces. Once they're so close together and they are the synapse is complete, then crossing over can begin. And so that's where the 2 chromosomes actually, the 2 non-sister chromatids of the 2 chromosomes are going to exchange genetic information. Now, when that happens, it's not like you can really see that happening in real time. So what you do see after it happens is these structures called chiasmas, or in plural chiasmata. And these are the sites of the crossing over, and they're actually visible. It almost looks like scars on the chromosomes. Now, when we talk about this and we show in pictures and whatnot crossing over, we always show like 2 different colors. You know, like here you have the blue and the orange chromosomes to try to demonstrate this crossing over and how the genetic information gets transferred. But you got to know, in reality, when they're looking at these chromosomes in a microscope, they're not going to be different colors. And so what they do see, though, is this chiasma, this scar, and that's how they know that crossing over has occurred. Once the chromosomes start to separate, just move slightly apart, the chiasmata actually kind of move down, and this is during diakinesis, towards the end of the tetrad, and that's called terminalization. Also, at the end of prophase 1, the nucleolus and the nuclear envelope starts to disappear, and other things that happen right before cell division, like the centromeres attaching to the spindle fibers and all that begins to occur at the end of prophase 1. But the main thing to take home information about prophase 1 is you have these chromosomes pairing up, these homologs pair up, synapse, and exchange genetic information. Okay, so during metaphase 1, that is where the chromosomes are now lined up along this equatorial plate. And the way that they line up is completely random. So here you see like, I'm not sure which is which, but we'll say the red's paternal and the blue is maternal. So the red, you've got a paternal one on this side, a maternal one, and another paternal one on this side. You've got 2 maternal and 1 paternal. And then you can see here where they've exchanged genetic information. So that's all random. The terminal chiasmata are still holding those tetrads together. Okay, so like I was saying, the alignment is random. This is the basis of independent assortment, which is something we'll talk about in more detail in Chapter 3. But basically this means that all the different alleles that are present on these chromosomes have an equal opportunity to end up, at one pole or the other. You got a 50-50 shot. So, for example, if your mom was a carrier for cystic fibrosis and you've got a 50-50 shot of being a carrier yourself. And like I said before, note that you have a mixture of maternal and paternal chromosomes lined up towards one pole, and that arrangement is completely random. So anaphase is when the 2 homologous chromosomes get pulled apart. Once they're pulled apart, they're referred to as dyads, and that process is referred to as disjunction. Now, in some cases, a non-disjunction can occur. This is an error that occurs during meiosis, and it can lead to chromosomal abnormalities like Down syndrome. And when we talk about chromosomal abnormalities, we'll talk more in detail about non-disjunction and how that works. So each dyad is going to be one of the homologous chromosomes. It's going to be composed of 2 sister chromatids held together by a centromere that is holding the 2 chromosomes together via that cohesin, which again is protected by the Shigoshin, just like in mitosis. During telophase 1, it just depends on the species and the cell type as to whether or not a nuclear envelope forms around the dyads. It may or may not. The cell may enter some kind of short interphase, minus the S phase. There won't be any DNA replication that occurs between meiosis 1 and 2, and the chromosomes remain condensed. In some cases, the cells may go straight into meiosis 2. And start the second separation, the second cell division. Okay, so this is just showing you meiosis 1 in review. So you have the 5 stages of prophase 1, and then metaphase 1, anaphase 1, telophase 1. And then now we're going to go into meiosis 2, which starts with prophase 2. So meiosis 2 is almost identical to mitosis. The only difference here is you have half the number of chromosomes. So instead of 46 chromosomes being separated at their centromere, the sister chromatids being separated, now you only have 23. So during prophase 2, you have the chromosomes being attached to spindle fibers. During metaphase 2, those chromosomes are then moved to the center of the cell, that equatorial plate in the center of the cell. The Shigoshin complex is degraded so that the 2 sister chromatids can separate. During anaphase 2, the sister chromatids are pulled to opposite poles. Once the individual daughter chromosomes are visible, they're called monads. And then during telophase 2, you have a haploid number of chromosomes at each pole. The nuclear envelope forms around the chromosomes, cytokinesis occurs, and you have formation of your haploid gametes. So in review, at the end of meiosis 2, you have 4 gametes, each that have a haploid number of chromosomes. Whether or not those gametes contain maternal or paternal chromosomes of the homologous pair is completely random. And each monad has potentially a combination of maternal and paternal genes thanks to the genetic shuffling, the crossing over that occurred during prophase 1. And all together, this increases the level of genetic variation. Okay, so this is another animation that's provided with the book that goes over everything that occurs in meiosis, like we were just talking about. And again, I will load this on Moodle as a flash file, Shockwave flash file, so that you can look at it on your own time and read through the slides. So, interphase before meiosis, the chromosomes are in that relaxed stage of chromatin, and you can't really see the chromosomes. Once the cell starts to divide, that will change. So in prophase 1, the chromosomes have been duplicated, but you can't really see the sister chromatids yet, and the chromosomes, begin to condense down. At zygonema, you have the rough pairing that we talked about. The separation is only 300 nanometers apart, and they look like that's the bivalent chromosomes because you can't see the sister chromatids yet. And centrioles start to move to the poles of the cell. By the time we get to pachynemia, you can now see the individual sister chromatids. The chromosomes are paired more closely. They're only 100 nanometers apart. You can see these tetrads that are formed from the pairing of the homologous chromosomes, and the non-sister chromatids side by side are able to undergo crossing over. And so we'll see that happen here, and where those crossover events occur, the little scars that are left over are called the chiasmata. Now, at diakinesis is when the tetrads start to pull apart somewhat. You can see here they're kind of pulling apart, but they're still held together by those chiasmata. During metaphase, this is where we're going to have the homologs pulled apart from one another. And so at anaphase, you can see where the crossing over has occurred and how the chromosomes lined up at that metaphase plate is completely random. And so now at telophase, we have 2 individual cells with a haploid number of chromosomes, and the chromosomes are the dyads that still have their sister chromatids attached at the centromere. And like I said before, a nuclear membrane may or may not form around the chromosomes. It just depends on the species. So now we're going to get to the point where we're ready to do our second division. So we're going to have prophase 2, where the spindle attaches to the chromosomes again. And at metaphase 2, the chromosomes are lined up in the center of that cell. The 2 sister chromatids are pulled apart during anaphase 2. And then during telophase 2, you have the 4 individual haploid cells. The nuclear envelope forms around the new, what are now called monads. And you have the end stage of meiosis. Okay, so that's pretty much it for just the process of meiosis. The next thing we're going to talk about is how these particular processes occur specifically in production of gametes in males versus females in animals, so sperm versus egg cells. Okay.
Now what we're going to do is watch a video that connects two of this morning's themes, meiosis and sex determination. The question is, by what mechanism does an embryo come to carry a Y chromosome or a second X? The answer is found in meiosis, in the father. So in this video of human male meiosis, I want you to look for two things. Guess what they are? Gene swapping, the swapping parts of paired chromosomes, and dividing by 2, halving the number of chromosomes per cell. So you remember that human cells have 23 pairs of chromosomes, but to illustrate the principles, we're just going to show 6 pairs here, 6 pairs of chromosomes, and we're going to look in detail at 2 pairs. We're going to look at the XY pair here and a pair of autosomes. And you see that we have it arranged so that mom's chromosomes, right? You got half your chromosomes from mom, half from dad. We have it arranged so that mom's chromosomes are on the left in red. The chromosomes you got from dad are in blue on the right. And what's the first thing that happens in meiosis? The very first step is you actually don't divide by 2. You multiply by 2. You double all the chromosomes, double all the genes. So at the first step in meiosis is you go to 4n, to a 4n stage. That's where we're beginning. Let's roll the video. Okay, so we're now going to blow up and focus on this pair of autosomes on the left, and you see there that recombination can occur at any point along the length of this autosomal pair. Let's see what happens when recombination occurs at a particular point. There's the swapping. We've just swapped a bunch of genes. The same can happen on the other arm of that pair of autosomes. Now we're going to turn over to the sex chromosomes. That's where the SRY gene is located on the Y. And it turns out that the X and the Y can recombine only within, they normally recombine only within their ends. Let's see how the swap occurs in detail. So we've just swapped parts of some stuff from mom and dad down at the other ends, like so. So at this point, how many copies of each gene, of each autosomal gene, does this cell have? 4. We still have 4. We have 2 copies of the X and 2 of the Y, and 4 of every autosome. Okay. And what we're going to see is, what do we have to do now? We've got to go from 4 copies down to 1 per cell. So we're actually going to have 2 rounds of division. And you're going to see the X up here is going to go to 2 cells on the top. The Y is going to go to 2 cells on the bottom. We're going to get 4 sperm out of this 1 cell. Let's roll the video. Okay, here comes the first division. The cell on the top has 2 copies of every gene. The cell on the bottom, 2. Now comes the second division. Remember, we've got the X's in the 2 cells on the top, the Y's on the 2 cells on the bottom. And these are going to go on to mature into sperm. So we will now have X-bearing sperm on the top and Y-bearing sperm on the bottom.
So this slide just shows you kind of pairing up the difference between mitosis and meiosis. So in both cases, you start with a diploid cell. With mitosis in the end, you end up with 2 daughter diploid cells that are identical to each other and also identical to that parent cell. In meiosis, however, you have 2 divisions, meiosis 1 here and meiosis 2 here. Meiosis 1 is the reductional division, meiosis 2 is the equational division. With meiosis 1, the homologs are separated from one another, and in meiosis II, the sister chromatids are separated from one another. And in the end, you end up with 4 haploid gametes. Okay, so let's talk a little bit about development of gametes specifically in animal cells. So you have, in males, it's called spermatogenesis, and in females, it's called oogenesis. And they're different in males and females. With meiosis in males, you end up with 4 sperm cells called spermatozoa. But with each single meiosis event in females, you only create 1 single egg or ovum. Also, meiosis in males is continuous, and in females, it actually kind of occurs in what I call fits and starts. It'll progress a little bit, and then it'll stop at a certain stage, potentially for years, and then it'll start back up again. So it's very different for females. Okay, so this is just kind of showing you the difference between spermatogenesis and oogenesis in, side by side. So let's talk about them individually. With spermatogenesis, it takes place in the testes. The undifferentiated diploid germ cell, which is called a spermatogonium, will grow and mature into what's called a primary spermatocyte. And at puberty is when we start with these primary spermatocytes undergoing meiosis I to produce 2 secondary spermatocytes. And so this is where the homologous chromosomes have separated. And then the secondary spermatocytes then undergo meiosis II to produce 4 immature spermatids. And so these are just basic looking cells. They have to undergo a bit of maturing to become those specialized spermatozoa that have the little tail and have that characteristic shape of a sperm cell. And so they will mature into specialized spermatozoa. So with each primary spermatocyte, you end up with 4 spermatozoa. And this is something that just continuously occurs through the lifetime of a male. And a 14-year-old boy is just as likely to fertilize an egg as a 80-year-old man. Now with oogenesis, it's completely different. This takes place in the ovaries. The daughter cells that are produced during the division, both the meiotic divisions, meiosis I and II, you end up with unequal amounts of cytoplasm. Really, one cell gets all the cytoplasm and the other one just dies. So the cell that receives all the cytoplasm eventually becomes the egg. The ones that don't become what we call polar bodies. They're basically just a cell with a nucleus in it, and they die. They may or may not divide again, but they don't develop any further. And meiosis is not a continuous process in females. Like I said, it kind of goes in fits and starts. So just to go through this process, you have that undifferentiated oogonium, which grows and matures into a primary oocyte. These primary oocytes are present at birth. So this occurs during fetal development, and a female is born with a finite number of primary oocytes. So when puberty begins, 1 primary oocyte will undergo meiosis I to produce 1 secondary oocyte and a polar body that pretty much dies. It may or may not divide again, but if it does divide again, it disintegrates. It doesn't become an egg or anything. So the secondary oocyte that gets produced is then released, and that process is called ovulation. So this occurs once a month for the most part. 1 primary oocyte will become 1 secondary oocyte, and that 1 secondary oocyte gets released through the process of ovulation. Now, only if that secondary oocyte gets fertilized, it will then undergo meiosis II. And actually, if you're doing in vitro fertilization and you're looking at it under a microscope, you can actually see that second polar body form, and that is indicative of fertilization being successful. So only if a sperm fertilizes that secondary oocyte, it'll then undergo meiosis II, produce an ootid that matures into an ovum, which is considered the egg, and will produce that second polar body that disintegrates. So you can see here you start with one primary oocyte and you end with one egg cell. And you don't even get to that point unless that egg cell has already been fertilized. Now in plants, it's very different, and I'm not going to go into detail about this. This picture's in your book. I just wanted to throw it up there to show you that their process is completely different because plants can actually fertilize themselves. They have both the male and the female parts present within the flower here, and and they can. So it's completely different. We may later in the semester talk more about plants, but right now we're just going to stick mostly to animal cells. So I believe that is pretty much it for this lecture. So we're finished with chapter 2. You can go ahead and finish this lesson and answer the rest of the questions associated with chapter 2. And we will be finished for the week. You can always come back and watch these videos again, or you can pull up the whole PowerPoint online without me talking over it, so you can go through the slides at your own pace and without me drawing all over them. And you can also print them out, PDF version, or you can print it out from PowerPoint if you want, but I also put a PDF version up there to make it a little bit easier for printing out slides. So, and always, if you have any questions about anything I've gone over, please feel free to contact me. And that's it. Signing off.